All right, hey guys, so it's 9 o'clock. I'm gonna go ahead and start. But before I do want to let you guys know that I did make a few mistakes on my PowerPoints that I posted on Friday. So yesterday, I usually on Sundays I go over them and make sure to correct any mistakes that I made. So I did that, and then I re-uploaded them last night. So as we're going through these, you may find some differences. So make sure to correct those. But if not, they're already uploaded with the corrections, so you can just re-download them. Another thing that I did want to talk about was, oh yeah, so I'm going to be posting my exam, my practice exam, sometime next weekend. So you guys have it for over the weekend for those of you who want to take it a little bit early. And then the TA schedule should have been posted. I posted it a couple days ago, so you guys should be able to figure out, what the schedule is for the next 2 weeks. All right, so if there's no questions, I'm going to go ahead and start the review session. We're just doing acid base part 2 and then the first GI lecture. Okay. So this is probably one of the more trickier kidney lectures, in my opinion. So I'll try to explain everything as clear as possible. Okay, so the first few slides are just review from the first part 1 acid-base lecture. So you've got your volatile acid, and the only example of your volatile acids that you need to know of is your CO2. Don't worry about these numbers, just know that your lungs remove a lot of CO2, so it's mostly your volatile acids that we are getting rid of regularly. Then you've got your fixed acids. These are going to be removed from by your kidneys, and your examples of these that you do need to know are your sulfate and your phosphate buffer, as well as ammonium chloride, and you should know lactate, but we don't really talk about it too much otherwise. Also know that sometimes lactate can be converted into CO2, so then it would also be considered a volatile acid that's removed by the lungs. Once again, don't worry about these numbers here. Just know the differences between the two. You can also ingest a fixed acid, and your kidneys would be responsible for removing those, such as salicylates, which are aspirin, for example. And other toxins can also lead to your kidneys' removal of those fixed acids. So you should be somewhat familiar with these examples. So once again, your total acid production, most of it is going to be volatile, as in CO2, and then a very small percentage of it, but still very important, is your fixed acids that are removed from by the kidneys and play a role in the production of bicarbonate. So moving on to this, once again, you should know the order in which your body tries to rebalance pH when your pH is disrupted. So your chemical buffering is going to be mostly your bicarb buffer. Your respiratory compensation is going to be the removal or maintenance of CO2. So depending on whether you want to make your blood more acidic or more basic, you're going to either hypervent or hypoventilate. And then renal compensation. So that's either reabsorbing bicarb or removing more bicarb, or also secreting protons to get rid of them, or inhibit removal of protons so that you don't get rid of as many in case you have more alkaline pH. Okay, so these are your 3 buffer systems that you should be familiar with already. You've got your bicarb buffer, which is the most significant one. This is the one that you find in your ECF. So we talk about this one a lot when we're talking about plasma pH. Your phosphate buffer is going to be the predominant one in your ICF. So this is the one inside of your cells that is doing most of the buffering. And then you also have your protein buffer. So this is going to be in your red blood cells. So specifically, an example of this one would be your hemoglobin. That we'll talk about in a few moments. But you should know these 3, and you should know where they are predominantly found. But for the most part, all of these are going to buffer your entire body such that they all have a pH of 7.4, because all of the compartments are going to equalize in pH because of the isohydric principle. So make sure you know that. Once again, he's reminding you of the Henderson-Hasselbalch equation. So you should know that as well, but I don't think he's gonna give you like any difficult math problems on that though. Okay, so we can run through this really quickly again. So in order to find the pH of your blood, you do need the Henderson-Hasselbalch equation. And since we're looking at your blood pH, you're going to be using your bicarb buffer and concentration. So this is the bicarb buffer base. So you have the concentration of your base, which we know is about 24 millimoles. So you should remember that number because that's important. And then we have, for our acid, we have CO2 and your carbonic acid. So remember your carbonic acid ratio of CO2 to carbonic acid is going to be 400 to 1. So obviously we have a significantly higher amount of CO2 in our blood compared to carbonic acid. So we can just kind of ignore the carbonic acid and find the concentration of CO2 instead, which is easy to find because we just need the partial pressure of CO2, which we know is about 40. And we can use this number to convert it to millimoles. So this is just a conversion factor. And then we can solve for the pH of blood, which comes out to be 7.4. Okay, so we kind of already learned about that, so I'm kind of racing through it. If he posts slides twice, it means he really wants you to know it. So make sure to focus on the slides that he reposted in the part 2 lecture from the part 1 lecture. So this is one that he's posted twice already. This is your isohydric principle. Once again, the different compartments. So inside your red blood cells, inside your cells, and inside your extracellular space is going to all have the same pH, so your different buffers are going to all buffer it to the same pH so that they're all equal to 7.4 under normal conditions. Oh, Kara, did he mention the pK of proteins? No, oh, sorry. Yeah, no. He just mentioned the pK value for your bicarb. So I wouldn't really bother memorizing 6.1. Perhaps not, because, oh yeah, no, he also mentioned the pK value for your, I think it's your phosphate buffer as well. So I think it was phosphate from the last lecture and also your bicarb. So those are the 2 pK values that he mentioned. But I wouldn't really worry too much about memorizing the pK values because I feel like he would give it to you. You're welcome. Okay, so moving on, you should know the special characteristics of your bicarbonate buffer. Once again, we have plenty of bicarb, so we have a high concentration. So we have more bicarb than we have CO2 or carbonic acid, and this is very convenient because we need plenty of bicarb to pick up any additional protons that we get into our blood to help maintain our pH of 7.4. Another thing is that our bicarbonate to CO2 ratio is 20 to 1. I would maybe memorize that ratio. And that's important because once again, we want to make sure we have plenty of bicarbonate to pick up any extra protons. And we are also breathing out CO2 continuously, so it's not ever going to really build up beyond the concentration of bicarbonate for that reason. And then lastly, oh yeah, so it's an open system because CO2 is being breathed out to the lungs, so it's considered an open system. So we can control the amount of CO2 fairly quickly through the action of the lungs. Once again, the Henderson-Hasselbalch equation, make sure you know it. Okay, so moving on to the newer stuff, I'll kind of slow down a little bit since this is like, new stuff. This is your Davenport diagram. You should be able to read this and understand it. I don't remember if he had any graphs on the exam, but just in case, make sure you understand this Davenport diagram. So up here, we have our proton concentration, and down here we have our pH. So they are inversely proportional to each other. So as your proton concentration increases, your pH is going to decrease, so that makes sense. And then on your y-axis, you have your bicarbonate concentration. So each of these lines represents a different PCO2. So we're also considering the concentration of CO2 in this graph. So if we have a low CO2, we would expect our blood to be more alkaline. So here's our normal buffering line. So this is typically how well our bicarbonate buffer works by itself. So this is typically where we sit at our pH level and our bicarb concentration when our PCO2 is really low. So as you can see, our pH is significantly higher from normal, and our bicarb concentration is decreased. So this makes sense because if you have a PCO2 level or CO2 is decreased, you would expect the equilibrium to shift to the left to make more CO2. So you would also expect there to be a low bicarb concentration. Oops, that's a 3. So that's kind of how it works here. So this Davenport diagram is showing you your blood pH before like renal or lung compensation. So you're just really taking the buffer into consideration here. So that's why your bicarb would decrease as well under these conditions. So if we're looking at a higher PCO2, so let's say 80 here, so the opposite would occur. So you would see an increase in CO2 would shift the curve, shift the equilibrium to the right. So you would also expect there to be higher levels of bicarb as well, which is shown here. So remember, 24 is about normal, and that's, you know, lines up with our normal value of 40 millimeters of mercury for carbon dioxide. So as you can see here, when our CO2 is higher, our bicarb is also going to be higher for that reason. And as expected, you would also see a drop in your pH because of how much CO2 is added. So a rise in CO2 is going to drop your pH, and it's also going to lead to an increase in bicarb. Okay, so, and then the opposite, the reverse is true for a low CO2. So just remember these relationships, and you should be fine. So this is just our normal slope of our bicarb buffer, and you can see that it's going to, you know, it lines up with all of the lines here, so that's pretty much how we determine it. And I think that's all I would take away from this slide. So moving forward, now we're looking at this red line here. So this is our normal buffer line, the black one. The red one is showing a stronger buffer system. So as you can see, when you compare, let's look at PCO2 of 60. So when you compare, oh, just kidding, not 60. Let's look at 80, because I already have the number here. Okay, so when you compare the black line where it crosses, so this is where it's functioning at PCO2 of 80, your pH is going to be about 7.18. But for the red line, it's going to cross at PCO2 of 80 at about 7.2. So you can see right away that your pH is going to be higher when your buffer system has a steeper line. So just keep in mind that if you're looking at these Davenport diagrams and the line is steeper, it means you have a stronger buffer, meaning that the buffer will work better to maintain a pH closer to 7.4. So you can see that this steeper line at a higher CO2 level will make the pH a little bit closer to your normal level of 7.4. Okay, that's all that's really saying. And the opposite is true for a high CO2. So it's going to try to keep it as close to 7.4 as possible. And the steeper it is, the better it works at doing that. Okay, so this is showing you the same kind of concept, but now we're looking at it from hemoglobin. So hemoglobin is actually going to make your bicarbonate buffer work better by increasing your bicarbonate concentration. So you'll see here that as your bicarbonate concentration increases, so it starts off at 5 and then goes up to 20, your line is going to be steeper. So this is kind of the same. This is the same like values as before from the Davenport diagram. You have your pH on the x-axis and your bicarb concentration on the y-axis. So it's the same idea. So you'll see that there is a relationship here between hemoglobin concentration and a steepness of this buffer line. So we're just looking at it from our normal PCO2 value of 40 now. So we don't have all of those lines crossing it like we did in the last diagram, but it's showing you the same exact thing. So here is the relationship here. If you have more hemoglobin, you'll increase the slope of the buffer line, and that will allow you to make more bicarbonate and therefore cause for a more efficient buffer system. So you're able to maintain your pH closer to 7.4 when you have changes in the CO2 concentration. So instances where you would see an increase in hemoglobin would be if you have a higher amount of red blood cells, so a higher hematocrit. He could state a question like that. So keep that in mind as well. Okay, so now he's showing you an example of how that works exactly, or he's comparing your bicarb buffer with and without hemoglobin present. So this first example here is going to show you what happens to your bicarbonate concentrations with the addition of CO2 when you do not have hemoglobin present. So this is no hemoglobin present. You add CO2 into the solution of, I'm assuming it's like water and bicarbonate. So as you increase CO2, as CO2 increases, as you move across the x-axis, you're going to see a slight increase in your carbonic acid, which makes sense because if you're increasing your CO2, it's going to increase your carbonic acid. So you'll see a slight increase here. However, you won't see much of an increase in your bicarbonate. In fact, it's going to stay at the same amount. So you won't see a change in your bicarbonate because, some of it's going to be buffering those extra protons, and some of it is going to be like just released from carbonic acid. So you won't see a change. So when you kind of plug that relationship into the Henderson-Hasselbalch equation, you increase CO2, you're going to lead to a decrease in pH. So that tells you right away that this bicarbonate buffer by itself is not super efficient because you will see a change in pH, and we want to minimize change as much as possible. So in this second example here, we now have a solution that contains hemoglobin. So when you add CO2, same idea. As CO2 concentration increases, you do see a slight increase in your carbonic acid, but you also see quite a large increase in your bicarbonate. And he explains the reason for that in the next slide. So as long as you understand that hemoglobin increases your bicarbonate concentration, you will be fine. But here's how it is shown in the Henderson-Hasselbalch equation. So if you have an increase in CO2 and you also increase your bicarbonate, the ratio is going to stay about the same, and so pH is able to be maintained. So you won't see a change in this pH value. Okay, so this is why it works, and you should know, be able to explain how it works. So this is kind of important. So remember that CO2 is going to enter your blood and be in your plasma, and that it can also enter your red blood cells. So you should probably remember this from the POEM lectures, where you can have CO2 just cross the cell membrane and enter the red blood cell. So when it enters the red blood cell, your red blood cells also have carbonic anhydrase to make it into carbonic acid, and then that carbonic acid is going to dissociate into bicarbonate and a proton. So the cool thing about this is that hemoglobin has a negative charge. So it is able to pick up extra protons and kind of sequester it so that your proton concentration will be decreased because it's getting picked up and absorbed by this hemoglobin protein. So that's essentially like making more bicarb, because when we think of bicarb, we have to take into consideration the fact that for each negative charge, you need to cancel it out with a positive charge. But if we're taking this positive charge and removing it by binding it to hemoglobin, you are essentially making more bicarbonate since we're removing the positive charge. So there is an increase in this negative charge here. And this is just showing you the chloride shift. So when you remove a negative charge, move it across the membrane, you need to trade it with a negative charge to maintain the membrane potential, so it doesn't like tip off the charge balance between the two. So that's pretty much what you need to understand about how hemoglobin can allow for increased bicarb. And it's really just by taking the protons for itself and effectively making more bicarb out of that. So make sure you understand that. That's kind of important. Okay, so this slide again, I'm going to skip it. Okay, so now we're actually looking at how your bicarbonate buffer reacts to the addition of a strong acid. So this is what happens when if you were to like drink hydrochloric acid or something like that. It's going to lead to acidosis. Obviously, it's going to drop your blood pH. So the first thing that happens is going to be your fast chemical buffering. This is pretty much considered the immediate response because it's just like the first thing that happens. So now he's showing you how to determine this through like chemical equations. So when you add 12 millimoles of hydrochloric acid, so this is pretty much just a given value, it's going to dissociate into 12 protons and 12 chlorides. So we're really just, concerning ourselves with the protons here. So those protons are going to get picked up by your bicarbonate. So remember, in our body, we have 24 millimoles of bicarbonate in our plasma. So it's going to combine with 12 of those. So we wind up having 12 millimoles of bicarbonate left because 24 minus 12 equals 12. And then you'll make 12 carbonic acids because you've combined 12 of these protons. So you can kind of associate your protons with carbonic acid since it's the same amount here. So those 12 carbonic acids are going to mostly dissociate into CO2 and water. So now our CO2 has increased by 12. So, so yeah, now you're adding 12 to our normal value of 1.2. So this is our normal value found in the blood under normal conditions. We're adding 12 to that, and this is our bicarb. So normally it is 24, but now since we're removing 12 of those, it becomes 12. So when we decrease this value and we increase this value, you expect your pH to drop. So this is the new pH, which makes sense because we are adding an acid, so we would expect our plasma pH to drop. I don't think he's going to make you do this math because I think he removed all the difficult math problems from the test. So don't worry too much about the math here, but you should understand the concepts. Okay, so looking at it from the Davenport diagram, since we are decreasing bicarb, since that bicarb is absorbing those protons and becoming CO2, we see a decrease in bicarb, so we would expect our y-axis to be lower. So we're looking at around like 14, 15. So that decreases, and then our pH is also going to decrease significantly. So our scale doesn't go all the way down to 6. So this is why it's, beyond the actual graph values. But this is where we would be on the Davenport diagram. Okay. So this is the second line of defense. So before your body starts to compensate for it, under normal conditions, since you have increased your CO2 concentration, you'll continue to breathe normally. So normal ventilation will be occurring because your body's pH response system hasn't kicked in yet, but through normal ventilation, you will be able to remove that excess carbon dioxide and get your PCO2 levels back to 40. So you're back at 40. You have normal CO2 levels, but our pH is still low because of the fact that we have lost our bicarbonate. So our bicarb has decreased, so we still have a lower pH. So now, through those mechanisms that we talked about in block 2, our lungs will start hyperventilating to lower our PCO2 beyond 40. So it's now going to be 25, and that's going to help lower or increase our pH by getting rid of CO2. So this is a good fast way of helping to raise our pH, but it's not sustainable. Our lungs can only hyperventilate for so long. So the best way to permanently fix the problem is through the action of the kidneys. So this is showing you what happens with ventilation. Our bicarb has decreased. It's still decreased because of the first step, when those extra bicarbs got converted into CO2, we breathed that CO2 out, and then we triggered the pH response to increase ventilation. So now our CO2 is actually below normal. So our pH has increased because remember it was like somewhere around here. So our pH has increased significantly. Our bicarb is still really low. It's still at the same place that it was before. So now we just have to fix our bicarbonate concentrations. So that's the last step. This is the slow renal compensation. Make sure you understand that this is going to occur over a period of several days. So it is more of like, it takes a while to really fix it, but it's more permanent. You can think of it as being more permanent. So your kidney is going to increase proton secretion, and it's also going to increase bicarb reabsorption. And those two actions together are going to get your levels back to normal. So these are the two normal values. This is your normal bicarb concentration, and this is your normal CO2 concentration. So together that will get your pH back to 7.4. So he will talk about the mechanisms for that in a few minutes. Okay, so this is just an overview of everything that just occurred. So you started off with low bicarb because those bicarbs picked up those extra protons from the hydrochloric acid. You dropped your CO2 levels due to the pH response from the lungs. So that increased ventilation, so you got rid of more CO2 to help increase the pH. And then this is where your kidneys kicked in and restored your bicarb by increasing reabsorption. And then once your blood pH went back to normal, your breathing will go back to normal, and you will restore your CO2 levels as well. So that will go back to about 40 millimeters of mercury, like normal conditions. So understand that process, just like the concepts. Don't worry too much about the math associated with it. Okay, so now we're looking at the different transporters in the kidney that are responsible for acid secretion and bicarbonate reabsorption. So these are the two mechanisms through which your kidney will either increase the pH or decrease the pH depending on what your body needs. So I've highlighted these, color-coded them based on where they're found, because you kind of put one cell and put all of these like transporters here, but they're actually found on different cell types. So this is the blood side. So this is what's getting reabsorbed. If it's going in this direction, it's getting reabsorbed. If it's going in this direction, it's being excreted or secreted. Okay, so the pink is showing you the transporters that are found in the proximal tubule. This should look familiar because remember, sodium is going to be transported with bicarbonate together because sodium pretty much transports everything in the proximal tubule. And we also have a sodium proton antiporter, which should also look familiar because that's also found on the proximal tubule, and we kind of briefly discussed it. And then these ones in blue are the ones that are found in the late distal tubule or the collecting duct. So this is just your proton pump. your potassium proton pump, which is also an ATPase, so keep in mind these require ATP. And then you have this bicarbonate chloride antiporter here as well. So make sure you differentiate which ones are found in which part of the kidney. Yeah, so these are going to be responsible for actually like excreting protons, because remember your late distal tubule and your collecting duct are actually going to have net excretion of acid. And then these here, this one and I believe this one too, are more so responsible for reabsorption of bicarbonate in the proximal tubule. So as long as you can like categorize that your proximal tubule reabsorbs bicarb mostly, and then your distal tubule and collecting duct are going to secrete acid. Those are the main functions of those cells in those regions. Okay. So this is showing you this exact just same thing that I just discussed. So your collecting duct has your proton pump and your potassium proton pump, and then your proximal tubule has your sodium proton antiporter, mostly involved in bicarbonate reabsorption. So this is just showing you one of the old slides that he's already mentioned before. This is the antiporter that we're discussing in the proximal tubule, so it should look familiar. Okay, so this is the cell type in the proximal tubule. Remember, this is just going to have your sodium bicarbonate transporter on the blood side of the cell, and it's going to have your sodium proton antiporter on the lumen side of the cell. So the sodium proton antiporter is going to absorb a proton, and at the same time, it's going to exchange it for, it's going to absorb a sodium ion, and it's going to trade it for a proton. So you're pumping out a proton into the lumen. So this is the tubular fluid that would ultimately become urine. So this proton is going to bind to the bicarbonate that has been filtered. So this is, upstream you have your glomerulus with your glomerular capillaries, your efferent and afferent arterioles. They're going to take out some of this bicarbonate. So this is the bicarbonate that you have filtered out at the glomerulus. It's going to bind to this proton and become carbonic acid. And so you also have carbonic anhydrase hanging out in the tubular lumen, and it's able to convert it to CO2 and water. So this is super important that you understand that this needs to be here, because otherwise you wouldn't be able to transport carbonic acid into the cell. It can only be transported as CO2, because CO2 can freely diffuse across, as can water. Without carbonic anhydrase, you won't be able to reabsorb bicarbonate. So when it enters as CO2 and water, carbonic anhydrase once again is present inside the cell, and it converts it to carbonic acid and ultimately to bicarbonate and a proton. So bicarbonate will then be reabsorbed through this sodium bicarbonate transporter back into the blood. And that extra proton is going to then be exchanged once again. So note that the proton gets excreted, it gets picked up, and then it ultimately gets reabsorbed. So you really just have this recycling system of using the same protons over and over again to reabsorb bicarbonate. So that's super important to understand, that you're not going to have any acid excretion at the proximal tubule, because these protons are being recycled over and over again. So you're not going to see any protons leaving the proximal tubule in the tubular fluid. Okay, so to summarize that, we have a net reabsorption of sodium, which we already know about, and bicarbonate, because they go together. We have no net secretion of protons because they are recycled to bring bicarb back into the cell. And you don't see a change in tubular pH because, once again, the protons are recycled, so you're not going to see an increase in proton concentration over time or anything like that. What you should know is that angiotensin II enhances the sodium proton pump, because remember, angiotensin II wants to increase sodium reabsorption. And by enhancing the sodium proton pump, that does lead to an increase in sodium reabsorption. And because you're also increasing the amount of protons that you're excreting, you're indirectly leading to an increase in bicarbonate reabsorption because you need those protons out in the tubular fluid to bind to bicarbonate and turn it into CO2 so that they can be reabsorbed. So angiotensin has an indirect effect on bicarbonate reabsorption as well. And then the other thing to note, and I would maybe remember this if you have time, that your transporters can become saturated. So if you have filtered out way too much bicarbonate at the glomerular capillaries, you won't be able to reabsorb as much bicarb because your transporter proteins are going to become saturated. So you will see loss of bicarbonate in the proximal tubule when the filtered load at the glomerulus is beyond 40 millimoles. So just keep that in mind as well. Okay, so moving on to the actions at the collecting duct in the late distal tubule. Once again, you have your volatile acids, which are CO2, and your non-volatile acids, which is your phosphate buffer and your, I guess it's sulfate. So these also get filtered out. You'll probably remember that at the proximal tubule, you will see some reabsorption, but now we're talking about the distal tubule and the collecting duct. So a lot of this has been reabsorbed at the proximal tubule, but some of it still stays in the tubular fluid and makes its way down to the distal tubule and collecting duct. And so this filtered phosphate buffer and the sulfate are going to be responsible for kind of picking up protons as they're pumped out. So your ATPase pumps out protons like usual. And these protons are going to be picked up by, in this example, the phosphate buffer. And it's going to make it such that, you know, the proton is stuck to this phosphate buffer. It's not going to be reabsorbed because there aren't any transporter proteins for this compound, the phosphate. So it's going to be stuck in the tubular fluid. It's going to be removed in the P. And so this is a way in which we can get rid of protons. So you require these two to help with acid excretion. But keep in mind, it's only going to do about 33% of acid excretion. So it's only like 1/3, only does about 1/3 of it. So your bicarbonate, when it comes in, so it's going to come in as CO2 and H2O, as we already saw. And then it's going to have a carbonic anhydrase, which converts it into carbonic acid. And so this proton here is going to be excreted permanently. So similar to how we saw in hemoglobin, since we're taking the proton away permanently, we're effectively making a new bicarbonate in doing so, because now it's not like linked to a proton anymore. And you're able to reabsorb this bicarbonate, and you're effectively increasing the amount of bicarbonate that you are making. So you're increasing the concentration of bicarbonate through this through the action of, you know, acid excretion. Okay, but more importantly is ammonium excretion. So 66% of your acid excretion is going to be based off of the production of ammonium. This is ammonium. And so it requires you to have plenty of ammonia in your tubular fluid to bind to these protons. So same kind of deal here, where you'll have bicarb being reabsorbed to make effectively a new bicarbonate ion. And your proton gets permanently excreted through this ATPase or whatever kind of transporter protein can pump it out. It's going to bind to ammonia, and ammonia is going to kind of trap it, so it's stuck in the tubular fluid and needs to be excreted. So this is most of your acid excretion is through ammonia, and in the distal tubule, your distal tubule and your collecting duct, you're going to see a very high proton concentration because this is where most of your acid excretion is occurring. Also keep in mind that your the cortex of the kidney is going to be breaking down glutamine. So remember, when glutamine gets broken down, it's going to release two ammonia molecules as it does so, because it gets broken down into, I think, alpha ketoglutarate. So it releases ammonia as like a byproduct of that. And so this is the ammonia that is going to play a role in moving or trapping acids. So ammonia can actually diffuse across the membrane really easily, but once it picks up this extra proton, it can no longer diffuse across because of that positive charge, and so it's stuck in the tubular fluid and forced to be excreted in the pee. Okay, so you really need ammonia and plenty of ammonia in order to excrete most of your acid. Without ammonia, you won't be able to excrete most of your acid, and so if you have like metabolic acidosis, this would not be a good thing. So this is a summary slide. Don't worry about memorizing these numbers, but you should understand the relationship between the proximal and the distal tubule. So your proximal tubule is going to have a high amount of proton secretion, so it's pumping out a lot of protons for the purpose of reabsorbing bicarbonate. So overall, you're going to reabsorb 3,900 bicarbs by pumping out 3,900 protons. So there is no net acid secretion because these are the same number. So you don't see any acid excretion, but you do see bicarbonate reabsorption as a result of that. Because you're not seeing any acid excretion, you would expect your urine to blood proton concentration to not be that high. And your pH is a little bit lower because you are still pumping out protons to bind to the bicarb, but it's not like too much lower. In your distal tubule, this is where you're going to see most of your acid excretion. You're only pumping out 500, but these are staying in the tubular fluid and being excreted out. So for that reason, you're going to see that the pH drops significantly because it's not really binding to bicarb. So there's no real bicarb in the distal tubule to help buffer this pH change, as you would see in the proximal tubule. And then also you're going to see a high gradient because this is where you're seeing all of your acid excretion. So you would expect your urine proton concentration to be significantly higher than your blood proton concentration here. So you do see reabsorption of bicarb because it occurs inside of the cell. Since you're removing protons, you effectively make more bicarb. And you also make more bicarb because of that same action. So don't memorize these numbers though. Okay, so moving on, these are really important that you understand these graphs. Once again, I don't remember if he actually had graphs on the exam, but make sure you can understand these just in case he does put a graph on the exam. So this is your CO2 concentration. So it's kind of like a flipped version of the Davenport diagram. So now your x-axis is your bicarb. The actual lines represent the pH, and your CO2 is now on the y-axis. So this is your normal CO2 level. So this is where that crosses, and this is your normal bicarbonate level. So that is where you normally sit at, which is 7.4, which is your normal pH. So we'll see how these change depending on whether you have respiratory acidosis or metabolic alkalosis or acidosis or whatever. Okay. So with respiratory acidosis, this is going to be considered anything that will increase your CO2 concentration. So you're not really thinking about your kidney at this point. You're just thinking about CO2. Don't think about bicarbonate either. So whenever you have an increase in CO2, you expect your pH to drop. So with acute respiratory acidosis, this is this line here. Obviously, your pH is going to be decreased. So this is 7.15 to 7.22. And your bicarbonate is also going to be it's about normal, I would say. I don't know. It's like a little bit lower, but just about normal for whatever reason. So then chronic respiratory acidosis, since it's chronic, now you're thinking about your kidney and taking your kidney into consideration. So it's going to allow it to shift to the right because now you're reabsorbing more bicarbonate. So your bicarbonate concentration is going to increase, and it's going to help minimize the drop in pH. But you still have a decreased blood pH, but it's not as bad as it was before because of the actions of the kidney, allowing for more bicarbonate to be reabsorbed and therefore buffering the blood a little bit better. So as you can see in the Henderson-Hasselbalch, you should be able to understand the relationships between these because he can ask you like in terms of like arrows, like what would increase and what would decrease. If you have an increase in CO2, obviously it's going to drop your pH, but when you take chronic respiratory acidosis into consideration, you will increase bicarb a little bit. So your pH decrease won't be as significant because of that. It won't bring it back to normal though. You should understand the kinds of things that can cause these, just like a generalized understanding. So COPD, CHF, anesthetics, and some drugs. He will likely say one of these in the question stem to hint that he's talking about respiratory acidosis. So make sure you kind of memorize these examples that he has. He's not going to throw something random out there that he's never discussed. So it's going to be one of these if he does ask a question about respiratory acidosis. Okay, so for respiratory alkalosis, now this is going to be caused by hyperventilation. So this is going to be a drop in CO2, which is going to lead to an increase in pH. So this is acute alkalosis, and you'll see that your bicarbonate levels are going to be slightly lower, or actually quite a bit lower than normal. This is going to increase your pH, so it's going to overlap with these higher pH values as well, depending on how bad it is. So the reason why your bicarbonate decreases a little bit is, once again, when we take the Le Chatelier's principle into consideration, when you have a drop in CO2, you're going to shift it to the left so that you also see a drop in bicarb. So bicarb will decrease a little bit as well. When you have chronic respiratory alkalosis, now your kidney is going to want to secrete bicarb because your blood is too basic, so it doesn't need as much bicarb, so you're going to reduce the bicarbonate even more than before to get it closer to 7.4. So it's still not quite at 7.4, but it's a lot closer than it would be at acute respiratory alkalosis. So as you can see, the kidney does play a huge role in trying to minimize the change in your pH when you're talking about respiratory acidosis or alkalosis. Another thing to take into consideration here is that he considers respiratory alkalosis to occur when your PCO2 drops below 35 millimeters of mercury. I would memorize the normal and abnormal values for these because he will give you like a table with abnormal values like he did in his practice questions that he posted, if you guys looked at those yet. But yeah, you should memorize like the normal value range and what's abnormal so you can help determine what the problem is in his questions, because those will be test questions. Okay, so he could also use these examples in the question stem to help you determine what you're looking for. So pain, anxiety, hypoxia, anything that is going to lead to hyperventilation will be leading to alkalosis, respiratory alkalosis. So if you want to look at it from the Henderson-Hasselbalch equation, you drop CO2, that will lead to an increase in pH because they're inversely related to each other. Okay, so this is probably more important than the respiratory stuff. He really does focus on metabolic acidosis quite a bit, specifically metabolic acidosis, not so much alkalosis. But so you should definitely know this slide and everything that causes metabolic acidosis. So with metabolic acidosis, now we're thinking about bicarbonate. So respiratory, we think of CO2 changes. Metabolic, we think of bicarbonate changes specifically. So it's not going to have an acute and chronic because it's, you know, its own thing. It doesn't have an acute version of itself because it's the kidney, and the kidney is like slow. So a metabolic acidosis is going to obviously lead to a drop in your pH. So it's going to be one of these values. It overlaps with one of those values. And you'll also see a drop in bicarbonate because it's metabolic acidosis. So you expect there to be a decrease in bicarbonate. So your CO2 levels are also going to decrease likely due to the fact that same kind of relationship here. If your bicarb levels decrease, you would expect there to be a shift in the equilibrium to try to make more bicarb. So that's also going to decrease your CO2 levels as well because of that relationship there. So on top of that, I think this is prior to respiratory compensation because your lungs will start to hyperventilate to try to get rid of some of that CO2. So that could also be contributing to a drop in CO2 in metabolic acidosis as well. Okay, so the causes of metabolic acidosis are going to be anything that leads to a loss of bicarbonate ions. So his main example that he does use is diarrhea. So make sure you know that diarrhea leads to a loss in bicarbonate ions. Renal dysfunction, he may say, but that's kind of vague. So he would most likely be more specific about that. So anything that leads to an inability to secrete proton ions. So if your proton pump is, inhibited, then you will see a little bit of metabolic acidosis because of that. Ketoacidosis will also lead to that. So diabetes, ketoacidosis, or starvation, he'll likely use diabetes ketoacidosis as an example. Lactic acidosis can also cause it. That's also going to lead to increased acids in the blood, or anything that is a drug that's acidic can also lead to metabolic acidosis. Ammonium chloride, salicylates, ethylene glycol, methanol, or ethanol. I don't know which examples he would use for these, but I would just memorize all of these examples just in case as being types of drugs that lead to metabolic acidosis to help you figure out what it is in the question stem. Okay, I guess that's all. Keep in mind that all of these, so by decreasing bicarb, if you have an inability to secrete proton ions, the reason why bicarb decreases is because more bicarb is going to be used to pick up these proton ions. And so you're effectively removing bicarb in that sense because you're converting it to carbonic acid. So that does lead to a bicarb drop. So you can think of it that way if that's a little bit confusing. So all of these are going to lead to a drop in bicarb. Okay, so metabolic alkalosis, the examples he has here are either ingesting bicarbonate directly or antacids as well. Anything that's just going to add base to your plasma or your blood, it's going to make your blood more basic. And so you wind up with a lot of bicarb as a result of that because these bases are going to go and absorb acids. And so that's going to kind of lead to an increase in bicarb because now bicarb doesn't have as much acids to absorb. So that's going to increase your bicarb concentration. And then loss of acid as well. So if you have a drop in protons, that's going to lead to more bicarb than protons. So you lead to an increase in bicarb as a result of that. So the example he will likely use is vomiting. So all of these other ones you can kind of memorize if you have the time, but vomiting is the main example that he would likely use. So looking at the graph here, metabolic alkalosis is obviously going to lead to a higher pH, so it will likely cross the higher pH lines here. And you'll also see an increase in bicarb because of what we just discussed. So your bicarb concentration is going to increase. Your CO2 concentration will also increase because if you increase bicarb, it's going to shift it to the left. So you will also see an increase in CO2, not to mention the fact that you will see some respiratory compensation. You will be hyperventilating to get rid of some of that CO2. Oh, wait, yeah, no, never mind. It would lead to, it would be because of the equilibrium shift. Because if you have alkalosis, then you would have respiratory hypoventilation. So you would also lead to an increase in CO2. Sorry, that was confusing me for a second. Okay, as compensation. So that's why it's a little bit elevated here for those two reasons. Okay, so here's your Henderson-Hasselbalch. You increase bicarb, it's the numerator, so it's also going to lead to an increase in pH, so those two are related directly to each other. Okay, moving on. This is a overview of everything. You will see with respiratory acidosis, you're typically going to see renal compensation. So if the issue is in your lung, you're going to see compensation in the kidney. And then the opposite is true for if the issue is like metabolic acidosis or alkalosis, you'll see hyperventilation. But if your kidney is functioning normally and the metabolic acidosis or alkalosis is due to the addition of you ingested a base or an acid, then your kidney will also step in and try to fix it as well. But this is kind of more of the fast response. So he does go into metabolic acidosis in more detail in the next several slides. So this is good. This is a good summary slide, mostly for your respiratory alkalosis and acidosis, as well as your metabolic alkalosis as well. But not so much for your metabolic acidosis because he does go into detail. So yeah, so mostly use this for review of these three because that's all he really wants you to understand for these three examples. So moving on to metabolic acidosis, your kidney does play a role in trying to get your pH back to normal when you have metabolic acidosis, and it does so in three specific ways. So keep in mind, your lung will try to compensate for it a little bit, but the kidney is going to do the best job at trying to fix the pH. So this is why you will also see renal response in metabolic acidosis. So you'll see a decreased filtering of bicarbonate. So this is going to lead to less bicarbonate being filtered out at the glomerular capillaries. He doesn't really discuss the way in which this occurs, so I would just memorize this as a fact. If you filter out less bicarbonate, there's more bicarbonate in the blood, and so less of it makes its way out to the kidney, so you don't have to go through the trouble of reabsorbing it when that occurs. So it's just a better way of keeping bicarb in the blood. These two he does go into detail, so these are the ones that you really need to know. So if you increase your proton secretion in the proximal tubule, that's going to allow for reabsorption of bicarbonate. And if you increase your proton secretion in the distal tubule or the collecting duct, that's going to lead to a decrease in your proton concentration, which will also help with metabolic acidosis. And lastly, increasing ammonium production. So ammonia, remember, that's going to be like the shuttle bus that picks up the protons and causes it to be excreted out in the urine. So you need ammonia to remove your protons. So you would definitely need that to increase when you have metabolic acidosis and you're pumping out more protons, trying to remove more protons. You need more shuttle buses to pick those up, so to speak. Okay. Okay, so moving on. So this is just showing you that the difference between ammonia and your phosphate buffer, what they do under normal conditions. So normally your acid secretion is only about 100 because your blood pH is normal, so you don't need to increase acid excretion or anything like that. So remember, this is like 66%, and this is going to be about 33%, which that looks like the correct ratio here. So 33, this is like 1/3, and this is like 2/3 here. So that's your normal ratio of acid excretion. So when you do have acidosis, you will increase the amount of the phosphate and sulfate that is being excreted out with it. So it increases quite a bit, but your ammonia is the one that increases the most. So it's significantly more important when you are dealing with acidosis compared to your phosphate and your sulfate buffers. So this is the main one that we want to deal with when we're thinking about correcting for metabolic acidosis. So you should understand the things that are going to stimulate proton secretion. So obviously acidosis, any type of acidosis, metabolic or respiratory, are going to stimulate proton secretion because your kidney is going to want to excrete more protons when it wants to put your blood pH back to normal. So increase your blood pH back to 7.4. So those are going to stimulate proton secretion. These are going to kind of work together. So they're indirectly related to each other. And both of them are also going to stimulate acid secretion, even though they are kind of more ambiguous. Like you can't really think of it as, like it's not as obvious, I guess. So aldosterone is going to upregulate your proton pump, and that's going to help pump out more acid. And then hypokalemia is going to allow for reabsorption of more potassium so that you wind up pumping out more protons because of the potassium proton pump. So we'll look at that. This is probably the more complicated concept of this entire thing. So he doesn't go into great detail as to how all of this is connected, but like I looked in the book and tried to connect everything. I did have to change this slide because I wasn't correct about a few things. So this is one of the slides that I altered in my slides that I fixed and then re-uploaded. So it may look different if you haven't gotten the new version of it. Okay, so aldosterone is going to upregulate a few different transporters. So it's going to upregulate your sodium potassium ATPase. We saw that in one of the first lectures. And when you upregulate your sodium potassium ATPase, which I've shown here, you're going to reabsorb more sodium and you're going to pump out more potassium. So by doing so, it's going to lead to hypokalemia because you're pumping out more potassium. So we know that already, that it leads to hypokalemia. So when you have hypokalemia, remember that your cells have high amounts of potassium, whereas your ECF, like your blood, typically has lower amounts of potassium. So when you are pumping out more potassium into your tubule in your pee, so this is your pee, you're going to want to try to reestablish that potassium loss by moving potassium from your intracellular compartment to your extracellular compartment, right? So your proton potassium antiporter or ATPase, they're moving protons and potassium in opposite directions. So that's shown here. It's going to try to reestablish the potassium concentration in your blood by pumping more potassium out. And as a result of that, it's increasing the amount of protons that are pumped into the cell. Okay, so keep in mind that aldosterone is also going to upregulate your sodium proton antiporter and your proton ATPase. So now, because aldosterone is upregulating this ATPase, and this ATPase is also located here, it's going to start pumping out even more protons because more protons are coming in because you're moving more potassium out. So that's how hypokalemia indirectly leads to an increase in proton excretion by through that antiporter. So whenever you have acid excretion, you should always think connect that with an increase in ammonia production because you can't get rid of protons if you don't have ammonia there with it. So hypokalemia, because it leads to an increase in proton excretion, is also going to lead to ammonia production to kind of help get rid of more of those protons. So the opposite would be true. So hyperkalemia would do the opposite of that. So hyperkalemia is going to inhibit renal ammonia production for the opposite reason. So if you have hyperkalemia, you're going to be less inclined to move potassium back into your bloodstream. Because remember, with hyperkalemia and hypokalemia, that's talking about your blood concentrations of potassium, not necessarily like your intracellular concentrations or your tubular fluid. So don't get that confused. So with hyperkalemia, you don't want to move any more potassium into the blood because you already have plenty of potassium. So you're not going to lose as many protons because it's moving in the opposite direction. So that's going to inhibit ammonia production because you're not removing protons. So you're not going to need ammonia to help shuttle those protons out. Alkalosis will also do the opposite as well, because if you have alkalosis, you don't want to get rid of protons, so you don't need to make ammonia for it. Okay, so those are the ways that aldosterone leads to hypokalemia, and how hypokalemia can lead to ammonia production because it leads to proton secretion. So aldosterone indirectly leads to ammonia production. You can think of it that way. Okay, so lastly, the anion gap. So this is the anion gap is going to be equal to your plasma sodium concentrations minus your chloride and bicarbonate concentration. So so your cations and your anions need to be equal to each other. So you need to maintain the amount of protons and not protons, cations and anions, so that you don't mess with the membrane potential. So this is essentially talking about the concentrations of these in the extracellular fluid. So just think of it as your extracellular fluid concentration of sodium and chloride and bicarbonate and other negatively charged. So your anion gap is other negatively charged proteins or other things that contribute to the negative charge here. So when you lose bicarbonate, you need to maintain this equilibrium between your cations and your anions. So you're going to increase the amount of other negatively charged molecules. So if bicarb decreases in the ECF, so this is your ECF and this is your ICF. If bicarb decreases, it's going to need to get more chloride from the ICF and move it out to the ECF to help minimize this change in the charge between the two compartments. So it does that. So that's what the chloride shift is, and that's the purpose of the chloride shift. So under normal anion gap acidosis, when bicarb decreases, it's going to be replaced with the chloride shift. So in this case, HCl or diarrhea are going to cause normal anion gap acidosis because bicarb is going to be replaced with chloride, and that's how it works in that way. So you should memorize diarrhea and the addition of HCl as leading to a normal anion gap acidosis. Also, make sure you know this value here, the normal value that I've highlighted here, because you're definitely going to need to know that for the test, because he's going to give you those tables the practice questions that he posted. There's going to be like identical questions to those on the exam. So make sure you know the normal value for the anion gap. So when you have increased anion gap acidosis, now instead of increasing your chloride concentration, so your chloride is going to stay the same, you're increasing the anion gap. So the other negatively charged molecules are going to increase in the ECF by like moving from the ICF to the ECF or whatever. And this is going to occur with lactic acidosis, diabetes ketoacidosis, salicylate poisoning, methanol poisoning as well. So memorize these examples as causing an increased anion gap acidosis. And that brings me to my practice question from Friday. So I had originally made the whole, like the 4-part question about the kid that took ammonium chloride. I had originally used salicylate as the question. So when I sent that to Dr. Yip, he told me to change it to ammonium chloride for whatever reason. And so apparently ammonium chloride, because he didn't tell me to change it for the question number 4, where we're talking about the anion gap in that question. Apparently ammonium chloride would also lead to a normal anion gap. So that was like, I had left it as like salicylate, so it was increased anion gap, which was not correct. So just make a note in your notes that it actually causes a normal anion gap, and I was wrong about that on my exam. Okay, so I guess that's it for acid base. And these are the answers to these, in case you guys didn't get them. I don't know if he shared those with you. Okay, any questions before I move on to GI?
Phoebe, can you just really quick tell me why the chlorine moves into ECF one more time for the anion gap shift? Sorry, I missed it.
Yeah, no worries. So when you have a loss of bicarb, you're going to lose the negative charge in your ECF. And in order to maintain the balance between positive and negative charges, chloride from your intracellular compartment is going to move into your extracellular compartment to help like increase the total amount of negative charge and make it equal to the total positive charge. Does that make sense?
Yeah, this is not related to the chloride shift in the red blood cells, though.
Correct. Yeah, no, that's a different thing. It's the same concept because you do see movement of chloride to like minimize the change in membrane potential, but it's doing it to like equalize the positive and negative charges.
Gotcha, that makes sense. Thanks.
You're welcome. Okay, so a few questions. Are you going to review the 4 case studies you posted? Oh yeah, I can review those after the Kahoot if you guys want to stay, because I did do them. I just didn't want to do it before the Kahoot, just in case some people want to actually like attempt it first before I go over it. So yeah, I can do that after the Kahoot. And could you go over the aldosterone mechanism again? Yeah, that is tricky. So with aldosterone, it's going to lead to hypokalemia because you're pumping out more potassium out of the cells. So you're going to lose more potassium. As a result of that, your cells are going to want to pump out potassium back to your blood because of this loss due to aldosterone. And since they're doing that with the proton potassium ATPase, which is more of like, I guess it's like an antiporter, it pumps protons into the cell and potassium out, it's going to lead to more protons inside of the cell, which are then going to be pumped out because aldosterone also upregulates your proton ATPase and your sodium proton pump as well. So you see a loss in protons due to the hypokalemia. And so aldosterone indirectly through hypokalemia leads to an increase in ammonia production because when potassium is high, you're inhibiting ammonia production because you don't have a lot of protons being pumped out when you have hyperkalemia. So you don't need to make as much ammonia. And then when you pump out more protons due to hypokalemia, that will upregulate ammonia production. And acidosis does the same thing too. Does that make sense, Alexis? Or I'm sorry, Melissa. Okay, cool. Delaney, would an increased anion gap be any number over 16 or less than 8? So an increased anion gap would be any number over 16. So I think in his like practice problems, he uses like 25 or something like that. So it would just increase that number. Okay. Any other questions before I move on? Okay. If not, I'll go ahead and start the GI lecture, which is very long. Okay. So the first few slides, don't worry about the first few slides. It's really just like to introduce the concept of digestion. So we start off as single cell organisms that do phagocytosis, and then we need to maximize surface area if we're multicellular organisms. So you need to, in order to maximize surface area, you need to find a hollow shape, and that's why our GI tract is hollow, because it maximizes surface area, not to mention the fact that we also have microvilli, which increases the surface area even more for optimal absorption. Sorry, I'm like racing through this because it's not really important. So our GI function, you guys probably should know this already, like you digest your food to absorb nutrients. He also mentions that it acts as like a secondary nervous system, but I wouldn't worry too much about that because it is something that it's like a newer idea and they're still doing research on it. So it's just like a fun fact more than anything. But another thing to note about your GI tract is that because it is open to the environment, you do need to make sure that it has the components to minimize infection. So it will have to have like a barrier to toxins, bacteria, parasites, all of that. So that's something that he gives examples of as the lecture goes on. So he uses this like example. I didn't find it helpful last year. So you don't have to like look at it. He's not going to ask you any questions about the this like analogy. But you should be familiar with all of the components in this chart. You guys probably already talked about these in like anatomy and everything. So you're probably familiar with it already. Okay, so I'm just going to like go ahead and move on to the more important stuff. Okay, so the takeaway from this slide, it is quite a bit, but so you ingest food, it goes into your stomach. We know your stomach has a low pH. It does a little bit of absorption, but not too much. You can't really associate it with being like absorption. It's more of like digestion. Most of your absorption is going to be done in your small intestine. So you'll see that your heart pumps blood to your stomach, and it's going to give the stomach oxygen. So those cells need oxygen. So it's typically high oxygen, but as you move down the GI tract, you're going to see a decrease in oxygen because you're giving the cells oxygen. But the nutrients towards the end of the GI tract, the nutrients go up because you're absorbing more nutrients. So your venous blood is actually pretty high in nutrients. So that's, something I would just think about. And then your colon is going to be important for extracting water and reabsorbing electrolytes and that sort of thing. Okay, so you have your 3 phases of digestion. You've got your cephalic, your gastric, your early intestinal, and your late intestinal. The 2 that he talks about in this lecture are your cephalic and your gastric. So when you think of your cephalic, just think of it as like anticipation of food. So, the sight, the smell, even when you first put like a bite of food into your mouth, like that initial taste can also contribute to your cephalic phase. And it's going to lead to increased saliva and a few other things that will prepare your body for digestion. For your gastric phase, that is going to be where the food hits your stomach pretty much, and you start to increase acid production because your brain becomes aware of the fact that you just ate, so you want to increase other things to help absorb the food or break down the food. And then these 2 he'll talk about probably in today's lecture and Wednesday's lecture. So early intestinal is going to be when you release all those enzymes that help further break down the food in your small intestines. And then late intestinal is your colon, where you absorb H2O and electrolytes. So your cephalic response is going to be tied to your desire to eat. So the sight of certain foods will trigger your mouth to water, your stomach to secrete hydrochloric acid, and the rest of your GI tract to help prepare you to eat. Don't worry too much about this, just having a general understanding of your cephalic response. It's more of like a mental thing. You can think of it that way. More importantly is this slide here. So several different stimuli can trigger the cephalic phase. So auditory, for example, like he uses Pavlov's dog, like the bell will make the dog's mouth water, but like for us, maybe it's the sound of, I don't know, the toaster bell, because it kind of signals that you're about to eat something. Cognitive, like thinking about food, seeing food, smelling food, and then obviously tasting food will then signal to your higher brain centers, which then tells your DVC. This is an important thing to remember about your GI tract. It's like the only real big brain system that you need to know for your GI tract, I think. So that's going to signal parasympathetic signaling, obviously, and that's going to lead to your effects. So the effects include saliva, gastric, so you'll have more HCl, hydrochloric acid, pancreatic juice secretion, and then you will see mild contraction of the gallbladder, but keep in mind that you're not going to be actually secreting bile just yet because your sphincter of Oddi will stay closed. So that's something to probably memorize because it's a little bit different about the cephalic phase. Okay, so you probably already talked about salivary glands and histose, so this is mostly review. So your primary secretions are going to be isotonic. It's going to be mostly sodium. You'll see bicarb, chloride, potassium, those kinds of things that will be secreted from the primary secretions. And then your secondary secretions are going to happen in the duct. And this is where you're going to secrete even more bicarbonate and more potassium so that you reabsorb more sodium and chloride. So your final salivary secretions are going to be high in bicarb and high in potassium. So I would just memorize that primary is going to be the acinar cells, and your secondary secretions are going to be in the duct. But the next couple slides are probably more important in terms of like memorization. So this is your acinar cells. So this is like the bubble part of the gland. So these are going to secrete your isotonic fluids, your sodium chloride, but that's not super important right now. The important secretions that you need to know are potassium and bicarb, because that's going to make up the most of your saliva. Another thing to take away from the acinar cell secretions are the fact that alpha amylase, lingual lipase, and kallikrein and IgA are also going to be excreted from the acinar cells. This is your primary secretion. So attribute primary with acinar cells with these secretions here, and that is also isotonic. Okay. So you don't have to worry about memorizing all of these like little transporters. Just know this box and where it occurs, and you'll be fine. So your lipase is an enzyme that breaks down lipids. Your alpha amylase is going to break down starches. Kallikrein is a proteolytic enzyme. So those are the three. And then IgA should be pretty straightforward from immuno. IgA typically is going to be found in mucous membranes. So you're going to find it being secreted from your saliva to help protect against infections. Okay, so this is going to be your secondary secretions in your ductal cells. So in this area here, you're going to reabsorb some of that sodium that you had originally secreted in your acinar cells and the chloride as well. So it's no longer isotonic. You will start to excrete more bicarb and lots of potassium. So you should know these two things here and associate them with the secondary secretions of the salivary ductal cells. So you need lots of bicarb to make sure that your saliva is super basic, so that you can protect against tooth decay, because acids typically wear down your teeth, and the base of the bicarb can help decrease acid erosion. Okay, so he said you don't have to worry about the glands, so don't worry about the glands. Unfortunately, he didn't tell us that last year, so I actually memorized the glands, and I don't think it was worth my time. And then the function you should memorize and the general components. So the main ones that you should know are these enzymes. Those are like the super high yield ones. The other ones you can just kind of generalize. So like proline-rich proteins typically help with lubrication. Mucins will also help with lubrication. It's like the main thing that it does. These you should know that alpha amylase leads to starch digestion, lipase to fat digestion, ribonuclease to RNA digestion, and kallikrein to protease function. And then you've got all of these other things that are mostly going to help with like immune response. So they're antimicrobial and they can help you protect against infection. The one that he really just talks about is IgA, because he mentioned that in the last slide. So that's the one that I would say is worth memorizing. And don't worry about the region in which these are secreted, except for the fact that you should know that most of these are secreted, or these three are secreted in the primary secretions at the acinus, because he mentioned that previously. And don't really worry about ribonuclease. You can memorize it if you want, because it's like kind of like a different one. And he does want you to know it, so maybe memorize that one, but he didn't mention it specifically like he did the others. Okay, so chewing will help break down your food so you can increase the surface area available for further breakdown of the food bolus. So know what chewing does. Don't worry about the pounds of pressure. That's too much information, but maybe knowing that your incisors and your molars are involved. And that saliva is also important for chewing because it allows for lubrication so that you can easily swallow your food. Without saliva, it would be really hard to swallow food, especially if it's like really dry food. And it also secretes things that can start to digest it, like your alpha amylase, which starts to break down starches and that sort of thing. So it takes your chunks of food and turns it into a liquid bolus. So a bolus of food is what you will be swallowing ultimately. So that's the term for that after you've chewed it up. Okay, so this is still part of the cephalic response, is like gustation and olfaction. So you guys already know the 5 different tastes from neuro and how they work. You don't have to worry about that. So you don't even have to memorize this because you probably already have it memorized. Don't worry about this number. Just know that your cephalic response is going to be helped by taste and smell, and taste is dependent on these 5 receptors. Okay. So swallowing. He definitely wants you to know the term dysphagia. So dysphagia is going to be anything that causes it to be difficult or painful to swallow. And there's a lot of different things that can cause that. So a lot of them are neurological, since swallowing requires you to be able to like contract and like choreograph like contractions so that they all flow smoothly and you're able to push food down in like a perfect kind of way. So it does require your nerves to work well. So note that your swallowing reflex is coordinated by your medulla. So that's super important for that. And it also requires the appropriate sequencing of contractions of the skeletal muscle. So for you to swallow, you need to contract above the food bolus and relax below the food bolus to allow for movement downward. And also your sphincters need to be able to open up, otherwise you won't be able to swallow anything. So your medulla does that. So I would memorize that your medulla is involved in the swallowing reflex. And definitely no dysphagia because he underlined it. So that's something he definitely wants you to know. Okay, so this would be easy to remember. Since he underlined none, this is like the high yield thing he wants you to know. So the oral phase is like towards like your mouth, I guess. And it's mostly going to be striated muscle. Your esophagus is mostly made up of striated muscle at this point. So it's more voluntary. So anytime you see striated, you can associate that with more voluntary control. Your cortex and your medulla both like work together to do this. Remember, your cortex is more of like a voluntary thing. I think that's going to be your voluntary control. And then your medulla also helps with it as well. As you move down to the pharynx, so you're going to have more of like a pharyngeal. This is your pharyngeal phase. So the food bolus moves down a little bit more lower. It's still striated muscle. You do have some voluntary control because it is there is some striated, but now your medulla is mostly coordinated. You can't really think about contracting your esophagus once it's past your mouth. So that's pretty easy to remember. And then lastly, your esophageal phase. So definitely know the esophageal phase. And know that this is going to be a combination of striated and smooth. But since they're smooth, you can consider this to have no voluntary control, which makes sense because you can't really think about swallowing your food as it goes down past your mouth. So this is different because now your medulla is partially involved, but you also have local responses, which we'll talk about in the next couple slides. So I would say out of all of these 3, this last point is high yield because he does want you to know that there is no voluntary control in the esophageal phase. And if I vaguely remember there being a question about this topic here. So I would remember that specifically. Okay, so for a food bolus to go from your mouth down to your stomach, you're going to have peristalsis. So peristalsis does occur in your esophagus through the contraction of and relaxation of the esophagus in like a coordinated manner. But the first step that you need is, in order to get past it, you open up the upper esophageal sphincter and then down through peristalsis, it gets pushed down. And then lastly, in order for it to enter your stomach, it needs to open your lower esophageal sphincter, and your stomach needs to relax in order to accept the food. So you'll also see receptive relaxation. Once again, he's underlined these, so these are high yield and you should memorize these. So acetylcholine is going to be responsible for the contraction aspect of peristalsis, and nitric oxide is going to be the main molecule involved in the relaxation aspect of peristalsis. So Those two are super important for swallowing as well. Okay, so this is just showing you the same kind of concept here. It's a little bit more detailed. I wouldn't really worry too much about all of the detail in here, just kind of generalize everything. So this is your pharynx, this is your upper esophageal sphincter. So this is all under voluntary control because you have to think to swallow your food. So your glossopharyngeal afferents are going to send signals to your swallowing center, which is inside of your medulla, and tells it that it's time to swallow. So then your medulla is going to help coordinate the opening of your upper esophageal sphincter to allow food to enter. Note that all of this is striated muscle, so you do have voluntary control. And then past a certain point, it just becomes more involuntary and more involuntary until it's all smooth muscle, in which case it's like all involuntary. So there is like a transition here. It's not like super cut dry, you know, there's like more of a transition. So you use your somatic nerves when it's more voluntary and autonomic nerves when it's involuntary. So that's just what that's showing. That's like basic neuro. He's not going to ask you anything like that, but he does want you to know that there's like, it goes from striated to smooth and therefore becomes more involuntary. So once it gets to the stomach, it needs to pass this lower esophageal sphincter. So your diaphragm kind of goes across here. So your stomach is like rubbing up against the bottom of your diaphragm. So your lower esophageal sphincter kind of goes through the diaphragm. So that's what it means here. So you can get a hiatal hernia where your stomach kind of pushes through that little opening and you get some of your stomach poking out into your thoracic cavity because of that. So that's all it means by hiatal hernia, but he's not going to ask you about a hiatal hernia. He just wants you to know that the lower esophageal sphincter is at the bottom and that it needs to open in order to allow food to enter. Okay, so you should memorize the general pressure of the three regions. So your upper esophageal sphincter has a high pressure. So you need to actually put some effort into swallowing your food to push past the upper esophageal sphincter because it has a high pressure. But that also allows for once food get past the upper esophageal sphincter, that high pressure can help push food down as you swallow it. Your intraesophageal pressure is very negative, so that's going to help allow for food to be pulled along because of the low pressure in that area. So it makes it really easy for food to flow down. And then at the bottom, your lower esophageal sphincter isn't as high pressure as your upper esophageal sphincter because it does need to allow food to enter as it moves down through peristalsis. So I don't think peristalsis would be able to overcome the lower esophageal sphincter pressure if it was as high as the upper esophageal sphincter pressure. So it's not as high as a result of that. And so that's why you can get acid reflux because it's not as high pressure as the upper esophageal sphincter. But anyways, that is still high enough pressure to prevent reflux. But if there's any like deviation from that, it can, allow for reflux. Okay. So those are the three pressures. You should memorize those. Or like at least like know which one's the high pressure, which one's the low pressure. Okay. So you have a resting state, relaxation state, and constriction state of your GI sphincters. So if we're talking about your lower esophageal sphincter, which is the one he really focuses on, so we can just talk about it in that way. The resting state is going to be where the pressure in your sphincter is greater than adjacent segments, meaning it's not really going to allow food to come in. So this is just when you're just sitting down and not eating anything. You're going to be in the resting state. So there won't be any movement upwards. So this will help prevent reflux. But there won't be any movement inwards because you're not eating anything. So as soon as you eat something and you're swallowing it, that's when your pressure in your sphincter is going to be equal to the adjacent segment. So food is able to enter the stomach through that sphincter. So that's going to be the relaxed/open phase. And lastly, you're going to have the constriction/tightly closed phase. So this is going to occur when you're doing like when you're moving around a lot. Like, so when you're running, you're going to be tightly closed because you're obviously like jumping around a lot, and that makes it really easy for backflow to occur. So in order to prevent that, your GI sphincter, specifically your lower esophageal sphincter, is going to be tightly closed to prevent backflow when you're doing a high activity kind of thing. So in this case, your pressure in your sphincter is going to be significantly higher than adjacent segments for that reason. So since he underlined these, make sure you know the differences between them. Okay, so your lower esophagus, after you swallow, you're going to have primary peristalsis, which moves food down. If there is still distension in your esophagus, so your esophagus kind of closes in on itself, but if you have a little bit of food left after primary peristalsis, it's going to trigger secondary peristalsis because it tells your esophagus that you haven't swallowed all of your food properly because there's still some stuck in there. So it's going to trigger secondary peristalsis to help you swallow like whatever was left over. So this is something that happens when you eat something particularly sticky or something that's difficult to swallow. It obviously wouldn't really occur if you're drinking water or something more liquidy. All right, so this is an important slide. Understanding this mechanism is, I think, pretty high yield. So we talked about how the esophagus has, towards the bottom of the esophagus, we have both local and vagovagal reflexes. Both of these are going to be mediated by acetylcholine, which is going to lead to contraction, and nitric oxide, which helps with relaxation. He also mentions that vasoactive intestinal peptide, VIP, is also somewhat responsible for relaxation, but the ones he underlined are nitric oxide and acetylcholine, so those are the ones he really wants you to know. So this is going to be your vagovagal reflex here, so it's going to depend on your DVC. So this uses your brain here. So behind the food bolus, you're going to constrict to help push it forward. And so once it pushes it forward, it's going to sense a distension in your esophagus. And this is going to signal to the DVC. So this is a sensory or afferent sensory neuron. Signals to the DVC to then release acetylcholine upstream to help push it along, and to signal the release of nitric oxide to help relax it downstream, so it's more receptive to entering the dilated region. So that's what is going on here. These are just your myenteric nerves. They're just like the ones that actually release the acetylcholine or norepinephrine. So here's your local response. This does not require your brain at all. So there's a couple things that will trigger this. So distension would trigger it, like in your vagovagal reflex. But also low pH. He really doesn't discuss why low pH does this. Maybe if you eat something really acidic and it senses a low pH and thinks that there's food in there, maybe that's why. I don't know. But distension is the main one that he wants you to understand. So it senses distension or low pH, and that tells interneurons to go and release acetylcholine upstream of the food bolus to help contract and push it along, and then nitric oxide downstream, similar to this, but you just don't use the brain, you use interneurons instead. So same kind of concept, but just one uses the brain, one does not. So make sure you understand this to some extent. Okay, so dysphagia. Okay, so keep in mind this is difficulty or pain swallowing, and it can be caused by several different things. So GERD can damage your esophageal tissue, and that can make swallowing very painful and difficult. Achalasia is esophageal spasms. So if you have muscle spasms in your esophagus, it's going to make swallowing very difficult because it needs to be coordinated perfectly. Cancer can also lead to some kind of like occlusion in your esophagus that could also make swallowing very difficult. There are also neurological causes to this because remember, swallowing requires contractions to occur in sequence with one another. So if you have neurological problems that can cause that sequence to be interrupted, it's going to make swallowing very difficult as well. He also mentioned that if you were to get chemotherapy, that radiation can lead to damage of the salivary gland. So if you decrease saliva production and have like a dry mouth, it's going to make swallowing also difficult because you require that lubrication associated with swallowing and saliva to help move things down, which makes sense because if you eat something that's very dry, it's very difficult to swallow. Okay, so he mentions that your esophagus have receptors, chemoreceptors, thermoreceptors, and mechanoreceptors. I think this is like the more high yield concept, the pain with distension, and I vaguely remember it being a question, maybe that or like the cold pain. It was something about esophageal pain and what stimulant causes that, or something like that. But anyways, you have if you were to swallow something acidic, you have chemoreceptors in your esophagus, and it can cause some pain or like some burning, but it's questionable. So I wouldn't say he's going to ask you a question on this or anything. Yeah, and so we also kind of discussed that with like feedback control because your local response does respond to a low pH when you swallow something. So that can also act to help with peristalsis, as we saw in like 2 slides ago. Temperature is also going to be something that your esophagus can pick up on because it has thermoreceptors. But anytime you eat something that's really hot, it's going to burn your mouth. But when you swallow it, it doesn't hurt. You just sense it being warm, I guess. So it's a non-painful sensation. And then lastly, it's actually distension that can cause burning and pain. So if you were to swallow something that's really big, it's going to trigger the mechanoreceptors, because remember, mechanoreceptors respond to stretch, and something that's really big will distend your esophagus significantly. So that's actually going to cause burning and pain. And I'm sure we've all experienced swallowing something that's too big and then it hurts going down. Okay, I think it was something about cold ingestion being one of the test questions. So make sure you understand this. So it's weird that hot things do not trigger pain, but cold things do. So cold things do 2 things. When you swallow something cold, it's not only going to trigger pain, but it's also going to inhibit peristalsis. So that's something to just a fun fact to note, and something that I think he does ask on the exam. So make sure you know that. Okay, another thing that's kind of important is the factors that alter lower esophageal pressure. So now we're talking about the lower esophageal sphincter. So things that increase it are going to be anything that will promote prevention of backflow. So if you increase your lower esophageal pressure, it makes it less likely to cause acid reflux. So something that does this is an increased intra-abdominal pressure. So I wrote on my old notes that it was like a big meal. So that's wrong. It's actually any like exercise, which makes sense because if you're exercising, your lower esophageal sphincter is going to close more tightly, and that pressure is going to increase to prevent backflow during exercise. And then also an elevated gastric pH. So if you were to take antacids, it's going to not only minimize the hydrochloric acid, the pH, or increase the pH in your stomach, but it's also going to promote an increase in lower esophageal pressure to also prevent backflow. Proteins he mentioned verbally that also lead to an increase in lower esophageal pressure for whatever reason. So I would just memorize those 3 things as being factors that increase lower esophageal pressure. And then the next one is anything that decreases lower esophageal pressure. So lower esophageal sphincter pressure is going to decrease, and that would allow for acid reflux. So pretty much anything that causes you to have acid reflux, or that your doctor says not to eat. If you are experiencing acid reflux, are going to trigger a decrease in lower esophageal pressure, including gastric distension. So this is if you swallow a lot of air or if you eat a big meal, this is where you would see a decrease in lower esophageal pressure, which, I'm sure we've all experienced, like heartburn after a big meal. Okay. So GERD therapy, you should have a general understanding of this. So GERD is going to lead to esophagitis because you have acid reflux, and that acid is going to corrode your esophageal tissue. So because your esophageal tissue does have some mucus secretion to protect it, when you destroy that tissue, you destroy the cells that make the mucus. And that makes it even worse because now you have even less protection to the acid and the pepsin, and that further destroys your esophagus. If you have gastric reflux, they tell you to change your diet, so lifestyle modifications. Proton pump inhibitors are a way of doing that. And then he explained fundoplication. So that's kind of shown here where you wrap the top part of your stomach around the esophagus to increase that pressure to keep it closed at the lower esophageal sphincter. So just have a general understanding of this and the general therapies and you'll be fine. Okay, so moving on to the gastric phase of digestion. So when you think of the gastric phase, this is where he says to kind of put it. It's kind of ambiguous, like there's kind of a lot of overlap between cephalic and gastric phase. So for our purposes and for the purposes of the exam, associate it with, as soon as food hits your stomach. So so as soon as food hits your stomach, it's going to stretch your stomach a little bit. And when your stomach stretches, it's going to signal to the DVC that it has stretched. And this is going to lead to a vagovagal reflex, as we've seen with the esophagus, that leads to the release of norepinephrine and VIP. And remember, both of these are going to stimulate relaxation. So it allows your stomach to relax and accept more food into it. So it increases stomach compliance and also leads to the increase in acid secretion, which we'll see in a few slides from now. Another thing that he'll probably talk about in more detail in today's lecture is that as food starts to exit into the duodenum, that stretch, fat, and protein will signal CCK to be released from the duodenum, and that also allows for stomach relaxation as well as you're eating. So both of those kind of work together to help relax your stomach and prepare it for food entry. Okay, so your stomach also does churning, which helps it, move the bolus around and allow the acid and the enzymes to fully encompass the food bolus so you can properly digest it. So the mechanism is your muscle, but it is caused by slow waves that are made by the interstitial cells of Cajal, which he will mention, I think, in today's lecture. He introduces those formally. But anyways, your interstitial cells of Cajal are kind of like your pacemaker cells in your stomach. So this is your pacemaker region, and they send action potentials down to cause your stomach to churn the food around. So it's just like the pacemaker for stomach churning to help, move the bolus around and allow it for proper digestion and breakdown of the food bolus. So these are the 4 different regions of your stomach, and you should know them. The cardia is the top towards your lower esophageal sphincter. Your fundus is the top middle area. Your body is the middle part, and then your antrum is the bottom by the pyloric sphincter. Okay, so you should know this as well. It's very similar to the cardiac action potential waves. So it shouldn't be too hard to memorize. I guess I kind of color coded it, but because of the blue background, it's kind of hard to do that. But anyways, so whenever you have a resting membrane potential, you should associate that with an outward potassium current to help maintain the membrane potential. With depolarization, we have opening of the calcium channels, which is similar to the cardiac action potentials. And then following that, you'll have inactivation of the voltage-dependent calcium channels and activation of your voltage-gated potassium channels. So potassium is going to go out, and you're going to stop letting calcium into the cell. And then after a while, because you're still closing your calcium channels, some of them are still a little bit open, you'll have a balance between your potassium and your calcium at the plateau phase. And then repolarization is when all of your calcium channels have closed and your potassium channels are fully open. And so that will get you back to your resting membrane potential. So this is the case for all BER, which is going to occur in not only your stomach, but your small intestines and your large intestines. So you just have to memorize this once and you'll know it for the colon and the other intestines. So make sure you know these steps.
Hey, Phoebe, I was just a little confused on like the repolarization step for it because I would think that the plateau phase, like that last bullet point where he says activation of calcium gated potassium channels, wouldn't the like calcium gated channels be in the plateau phase and then the repolarization just be the potassium channels?
Yeah, I see what you mean. I think so during the plateau phase, I think what's going on here is that you have just started to inactivate your calcium channels. So you still have some calcium channels that are open because they're like significant before. And so you still have activation of your potassium channels. So some of your potassium channels are open. So as these are opening, these are closing. And so they balance each other out. And then at the end is when you completely inactivate all of your calcium channels. So I would assume that this happens right here, just right as it starts. So that's why it drops down so much. Does that make sense?
Yeah, I was just a little confused on like why he had that last bullet point talking about the activation of calcium gated channels. Because even if you look at like I think it's page 349 and 350 in the book, it talks about like the transient repolarization being the closing of voltage gated calcium channels, but then in the plateau phase, there's like another calcium channel that opens, and then the repolarization is just potassium. So I wasn't sure if like the slide was wrong or if I was misinterpreting the book or like really what was going on with this, you know?
Yeah, good point. That might be worth asking him about because he might have made a mistake. I think it looks exactly the same as it did last year, so I didn't catch that, but yeah, you might want to ask him about it so that he can like clarify if there is some kind of inconsistency with his slide.
Okay, I'll try to ask him about it I guess after class or during his review session.
Yeah, okay, cool. Alrighty. Moving on, you should know the general relationship between the BER in the three different segments. So your stomach has strong intermittent contraction. Since you're not pushing anything down a tube with your stomach, you're just going to do like your intermittent contraction to churn the food. So it's not as frequent. Your small intestine is going to want to push all your food down as quickly as possible because it's so long. So you're going to have way more frequent contractions and therefore more frequent BER and action potentials. So that's why they're so frequent. And then your colon, because now you're reabsorbing water and your food bolus is becoming drier and drier, it's going to need more strength to help push down that dry like bolus. I don't know what it's called, chyme. I don't know. So it's going to need stronger contractions, and therefore the action potentials are a lot taller, I guess you could say, because they are stronger. Don't worry about memorizing these numbers here, but you should understand the relationships between these three as I kind of described them, and that should be good. Okay, so I remember memorizing this whole thing, but I don't think I needed it. I think the important thing to take away was here and then just having a general understanding that you have a brainstem vomiting center that several different things can feed into and lead to vomiting. So Different things that cause vomiting are going to be different types of drugs or irritants. So these drugs, like your chemo drugs and any kind of toxin, can irritate your gastric mucosa, which can then signal through the vagus nerve to a chemoreceptor trigger zone, and then your NTS as well, to help trigger vomiting. Drugs and other hormones can also signal the chemoreceptor to trigger vomiting. Pharyngeal stimulation, so this is like if you were to touch your uvula or to stick a finger down your throat, that would also trigger vomiting. Pain sites, anticipation, or like some kind of like anxiety can also do it, because this is like more of like a higher brain center response, because it's more like you're thinking about something. And then if you're spinning, or like if you have vertigo, that's going to activate your labyrinth in your inner ear, and that's going to signal to your cerebellum to throw up. So have a general understanding of these. I would just know that the brainstem vomiting center is like the main thing here, because that's like the middleman of all of this. All of this would probably be too much information. However, he does want you to definitely know that prior to throwing up, you do want to protect your mouth and your teeth, and so you will increase salivation. Remember, saliva has lots of bicarb in it to kind of minimize acid corrosion, so you will want to make lots of bicarb before you throw up. You'll see also somatic muscle actions through retching, so that's like dry heaving will lead to retching, and that's prior to throwing up. You do see that. Suppression of respiration, so when you're throwing up, you're not actively breathing, which is a good thing because you don't want to breathe in what's coming out and lead to aspiration. And then lastly, you will stop the BER, so your normal movements of your intestines that stimulate the movement of food down the intestines will be stopped in preference for a different electrical activity that he doesn't go into, that are going to lead to retroperistaltic contractile complex. So he used this word, he underlined it, so make sure you know that retroperistaltic, it's literally what the name suggests, is to move food upwards and out. So know that vocab word, I guess. Okay, you guys know this from histo. He's not going to ask you any histology questions, so don't worry about this. He just wants to show you that your stomach has these like little inlet or gastric pits, they're called, and they're like little inlets to increase surface area. And then you have all of your different cell types in these little pits. So that's the only takeaway I would get from this, but you do have to know it for histo. Okay, so this is a super important slide. He doesn't introduce all of these different secretory products in the first lecture, but he will ultimately get to them. So once you learn all of these, this is a good summary slide for all of them. The ones he does talk about or introduce in this lecture are hydrochloric acid, obviously pepsinogen, mucus bicarbonate, histamine, gastrin, and just acetylcholine and somatostatin for those two. So histamine, gastrin releasing peptide, acetylcholine are all going to increase gastric secretion, whereas somatostatin is going to downregulate gastric secretion. So this is like the different one. And you should know the cells that these come out of and their function. So as you're going through this, make sure you use this as a summary slide to help you like reinforce everything. Okay, so carbonic anhydrase is also found in your gastric cells. This is your, I think it's your parietal cell. So your parietal cell is going to have carbonic anhydrase, which is super important for making protons. Remember, your parietal cell is going to be responsible for secreting hydrochloric acid. So this is going to occur through, use of CO2 that is made in the cell through the TCA cycle. And it's going to go through carbonic anhydrase with H2O, make carbonic acid. And then you're going to also reabsorb this bicarb back into the bloodstream, whereas this proton is going to be pumped out through the proton pump. This is the same pump that you'll see in the kidney, so it's really easy to remember it, and lead to secretion of acid and hydrochloric acid. So this is the chloric part of the hydrochloric acid. So together, HCl. And yeah. So make sure you know that carbonic anhydrase is essential for hydrochloric acid secretion. Okay, so here is your, I wrote gastric cell, but it's technically your parietal cell. Your parietal cell is the one that makes hydrochloric acid, so it has receptors for acetylcholine, histamine, and gastrin. And these receptors are your muscarinic, your H receptors, and your CCKB, which I don't think we've learned about yet. But anyways, you have these canaliculi in the parietal cell, and when the canaliculi, they can open up and increase their surface area to increase secretion of hydrochloric acid. And so this is regulated by acetylcholine, histamine, and gastrin, which together signal the movement of these vesicles to bind with the canaliculi, and then since they have the little proton pumps on them, they will allow for proton pumps to be present on the cell membrane. And this is done through either CAMP or calcium. I don't know, he mentioned that, but I don't think you have to know like the biochemistry of it. Just have like a general understanding that you would use these second messenger systems to help fuse the vesicle with the cell membrane. Okay, so this is just more detail about that. He mentioned somatostatin, histamine, acetylcholine, and gastrin. So these are the 4 that I would know from this slide. So histamine, acetylcholine, and gastrin, as we already discussed, are going to lead to hydrochloric acid secretion by binding the vesicles with the membrane and increasing the surface area to lead to acid secretion. However, somatostatin is going to do the opposite. It's going to inhibit secretion of hydrochloric acid. So it is going to inhibit the actions normally done by histamine, acetylcholine, and gastrin. So note that these 3 are going to work in a synergistic manner. So they work together to maximize hydrochloric acid secretion. So if one of these is not here, you're going to have a significant decrease in hydrochloric acid secretion as a result. I also mentioned here that your histamine is going to be G stimulatory mediated, so that's your GS, and then gastrin and acetylcholine are going to be using PLC. Once again, that's too much biochem, so I wouldn't worry too much about it. Just kind of know the general trend here. Okay, so this is actually a pretty good slide for connecting everything. So remember that your higher brain centers are going to trigger HCl secretion through different means, either through your cephalic response or your gastric response. Either way, it's going to lead to the release of acetylcholine. And these 3 cells all have acetylcholine receptors. So that's going to trigger your chief cell to release pepsinogen, your parietal cell to secrete hydrochloric acid, and your ECL cell to release histamine. So histamine will make its way to the lamina propria, and it's actually going to go and upregulate your parietal cell, as we saw in the last slide. Pepsinogen is going to be a unactivated peptide that will become activated when hydrochloric acid combines with it and cleaves it. And together it's going to lead to the increase in, hydrochloric acid leads to the increase in protons, and pepsinogen will be activated by your hydrochloric acid to make pepsin, which is a protease. So gastrin cells are also found in your stomach, but these are going to act as more of like a hormone. So they respond to GRP, which he hasn't really introduced yet, but he will. And when they are activated, they're going to release gastrin into the blood, and gastrin will also upregulate the release of hydrochloric acid through the parietal cell specifically, as we discussed. Okay, and so now talking about the D cell. So the D cell is going to do like negative feedback to make sure that you're not secreting too much hydrochloric acid. So what it does is it senses protons in the stomach lining, and when it senses them, it knows that the proton concentration is really high. So it's going to release somatostatin to inhibit G cell release of gastrin. So remember, gastrin will then go to the parietal cell and upregulate release of HCl. So in doing so, by inhibiting gastrin release, it's going to decrease the amount of HCl that gets released. So make sure you know the role of somatostatin as well. Okay, so this is kind of putting together your chief cell and your parietal cell. So your parietal cell has two roles. The one that we introduced today is its role in making HCl, but he'll later introduce that it also does release intrinsic factor, which is important for vitamin B12 absorption later downstream of this whole stomach stuff. So anyways, HCl gets released. Chief cells will also be upregulated when you have a meal to start secreting pepsinogen. Pepsinogen is going to be activated through HCl into pepsin. So the thing about pepsin is that it can also upregulate itself by cleaving pepsinogen into pepsin because it is a proteolytic enzyme. It can upregulate itself as well. But pepsin is going to be important for breaking down proteins because we can only absorb, I think, dipeptides at the most, or maybe it's amino acids. So we need to make sure we break down our proteins as best possible in order to absorb them. Okay, so this is just kind of combining everything together. Remember, your vagovagal reflex is going to not only lead to relaxation of the stomach so that you can accept more food coming in through like this distension mechanism. So these mechanoreceptors will sense that food's coming in. Some of them will upregulate NO release to help make the stomach more distended, as we already talked about. But some of this is also going to lead to the release of acetylcholine. Acetylcholine will then trigger the cells in the stomach, such as like the parietal cells, the chief cells, and the G cells, to increase HCl, pepsinogen, and gastrin release, as we already discussed. And then later down the line, or today actually, he will discuss about how your duodenum also plays a role in secreting gastrin to help increase your protons and pepsinogen release. So don't worry about this just yet. He will introduce this later today, I believe. Okay, so I made a flowchart to kind of connect all of those concepts together. Hopefully you'll find it helpful because it was like 3 slides and it's just very convoluted. So I added this for you guys. Okay. So lastly, you do need to know how you can limit stomach acid. He did underline them. He will ask a question about them. And I think I memorized the actual names, but they're very common, so it's really easy to memorize these. So you've got your antacids that are going to buffer your stomach acid and therefore minimize the amount of proton concentration available. So these are your Tums, your Maalox, and your Pepto-Bismol. So they're pretty common. Anything that blocks your acetylcholine receptor, remember, acetylcholine is going to be important for increasing your parietal cell secretions. So that's going to be inhibited or decreased when you block acetylcholine. So the only one that you need to know is atropine, which you probably already know from pharm. So, you already have it memorized. So that works out nicely. With histamine, now remember, histamine is also going to lead to your parietal cell releasing HCl. So these examples are Pepcid. That one's more common, so he'll likely use that one. I've never heard of Tagamet. Zantac is also pretty common, so he'll also likely use that. And Axid is another one I've never heard of, but yeah, maybe just memorize these two since these are pretty common already. And lastly, your proton pump inhibitors. So these proton pump inhibitors are specifically inhibiting your proton potassium ATPase in your stomach. And so these examples are Prilosec, Prevacid, and Nexium. So those are super common. And then these just directly inhibit the proton pump, so you can't pump any protons out, and therefore that's going to lead to a decrease in your proton concentration, and therefore your stomach acid. So make sure you know these. This is, a high yield slide. Okay, so I kind of drew out a graph of like how acid secretion changes throughout the day or between meals. So in between meals, obviously it's very low. So during the cephalic phase, you'll start to increase your amount of proton secretion. As you're eating, it's going to increase even more. And then as you're emptying it into the duodenum, that's when you'll start to see a decrease because you've finished eating. And then you start that cycle all over again. So if you can just visualize this graph, it'll help you remember this a lot easier. Okay, so in order to protect your stomach is responsible for secreting a mucus layer. This mucus layer is going to prevent gas HCl from coming down and destroying your tissue. So you need to make sure that your goblet cells, which are in your stomach, these are the ones that are responsible for secreting mucus. So these goblet cells are going to be secreting mucus. You've also got other cells that are going to be secreting bicarbonate as well. So it's a combination of bicarb and mucus that are going to protect your cells from acid corrosion. So if you have a decrease in mucus production, you're more inclined to get a stomach ulcer as a result of that. So Zollinger-Ellison syndrome is a syndrome he wants you to know is going to lead to a gastronoma, which is a tumor cell somewhere in your body. It doesn't necessarily need to be in your stomach, but it's going to secrete gastrin. Remember, gastrin is going to upregulate your HCl secretion, and that's going to increase stomach acid such that your stomach acid is greater than your mucus and bicarb production. Oops. And that's going to overwhelm your stomach and cause ulcers and stomach lining erosion and that sort of thing. So it's very dangerous. It can also inactivate lipase because if you are too acidic, it's going to denature the proteins, and these proteins, aka enzymes, are going to be inactivated as a result of that. So usually you can treat it with proton pump inhibitors or surgery. So know the treatments, know the cause. This is high yield, I would say. And then lastly, this is just a summary of like what the 3 different parts of your stomach are useful for. So the upper part is going to secrete mucus and bicarb. The bottom part is also just going to secrete mucus and bicarb. So these are both going to be like protective mechanisms. It's the fundus in the body, so the middle region, that's going to produce all of the other stuff like the hydrochloric acid and the peptides. So knowing what they secrete is very important. Motility, you can have just a general understanding of this and you should be fine. So obviously the top part is going to regulate food entry and anything going out. So preventing reflux is what it's important for, and also belching because it's going to regulate whether air can escape. It's also, the fundus in the body is going to be responsible for, forcing things downward. So it's going to have tonic force to help move things down. And then this is where all your mixing and grinding is going to occur. So you'll also see regulation of emptying into the duodenum as well. So just have a general understanding of the motility and make sure you do know the luminal secretions for these different regions. Okay, so that is the additional slides. You don't have to look at the additional slides, but because he's not going to test you on them, but they might be helpful if you're struggling with anything. So do you have any questions before I move on to the Kahoot? Okay, if not, I'll start the Kahoot. Sorry, I went over time.
Maybe while we're waiting for everyone to join, did you see that email about like the physiology class time being ended?
Yeah.
Okay, I wasn't sure if that was is that just like for a review, or is that for just the class is going to be longer?
So that's a good question. I got an email about it Friday, but like the email chain was like cut off, so I missed like the reason for it. So I actually have no idea, to be honest. I feel like it might be a review, but he's also having a review on Friday, so I don't know if it's like he's giving 2 reviews or if he's actually extending the length of class for whatever reason.
Gotcha. Okay, thanks.
Yeah, no problem. I'll give you guys about, I don't know, like a minute to join and then I'll start. Okay, I think I'll start it now. Okay, so at a higher PCO2, the equilibrium is going to shift to the right. So you would expect bicarbonate to increase as well. And your proton concentration is also going to increase because you have more CO2. CO2 increases, your proton concentration will also increase. Awesome. Yeah. So in order to correct your pH, you start with the buffering. That's the immediate response. If you have acidosis, you're going to want to hyperventilate to decrease your CO2. And then kidney reabsorption of bicarb would be the renal compensation mechanism. So good job. Awesome. Yeah. Ammonia is going to be the main way in which you secrete protons, and that's going to occur in the collecting duct or the late distal tubule. Okay, awesome. Yeah, so your proximal tubule has no net acid secretion, so you would not expect that to be more acidic than your collecting duct. Awesome. Yeah, normal anion gap is caused by HCl or diarrhea, and it's going to lead to more chloride being increased to make up for the loss of bicarb. Awesome. Yeah, that's the only one that's correctly matched to its particular cell. Awesome. Yeah, so you will see the reabsorption of sodium and chloride and the secretion of potassium and bicarbonate in the ductal cells. Awesome. Yeah, so somatostatin would inhibit it. Okay, so your antrum is towards the bottom of your stomach. That's going to secrete only bicarbonate and mucus. So the body of your stomach is going to be the one that's going to secrete all of the other stuff, including protons. Okay, so we're looking for all the stuff that decrease lower esophageal pressure, meaning you are more likely to have acid reflux. So gastric distension after a meal is going to cause that. Peppermint and alcohol are both going to cause that. Elevated gastric pH, that's like taking an antacid. So an antacid is actually going to increase lower esophageal pressure. All right, so I'm probably going to go over the practice questions he posted after this. So if you still want to go over those, you can stay. But if not, good luck studying, and I'll see you guys on Friday. Good job, whoever won. Okay. So let me just open up. Let's see. Okay. Okay. So I will go over all of these practice questions. Obviously not going to go over the true or false ones because he gives you guys the answers for those. So we're just going to go over these boxes, and you have to make sure you understand these perfectly because you'll get at least 2 questions on the exam that are literally exactly like this. He may change the causes. Maybe instead of like acute vomiting, he'll probably use like gastric suction, or it's the same idea though. You lose acid. Okay, so for the first one, the first thing we look at is the arterial pH. You should remember the normal values for these. So our normal value is 7.4. Here, actually, I'll make this into a slideshow. Okay, so our normal value is 7.4. So we know that this is below 7.4. So we know we are experiencing some kind of acidosis. So in order to rule out whether it's respiratory or metabolic, we then want to look at our CO2 levels. So our CO2 levels are low, meaning that it cannot be respiratory alkalosis or acidosis, because respiratory acidosis would lead to a high CO2 for respiratory acidosis. So we therefore know it has to be metabolic acidosis. So looking at our answer choices, which of these are going to cause metabolic acidosis? So acute vomiting means loss of acid, so that's going to lead to metabolic alkalosis, so we can rule that one out. Salicylate poisoning can lead to a drop in bicarb, which does lead to metabolic acidosis, so that is a potential answer choice. Diarrhea also leads to a loss in bicarb, so that also leads to metabolic acidosis. And then hypoventilation would mean that we have high CO2 and low O2. Here we have low CO2, so we obviously have to like cancel that one out. So now we have to determine the difference between salicylate poisoning and diarrhea. In order to do that, we need to look at the anion gap. So if we have a normal anion gap, we know that it's likely due to diarrhea. He could also use HCl ingestion as another example of one with a normal anion gap. So normal anion gap tells us, because our normal range is 8 and 16, so you need to make sure you memorize the normal range here. So we know that diarrhea is likely the cause because our anion gap is normal. So that is why diarrhea is the correct answer. For the next one, once again, first step is to look at the pH. It is high, meaning we're dealing with some kind of alkalosis. So to make sure it's respiratory or metabolic, we want to look at our CO2 levels. So our CO2 levels are high. A high CO2 would indicate respiratory acidosis. So we know right away that we're looking at CO2. CO2 here is a compensatory mechanism. So this is our lungs trying to compensate for alkalosis, meaning that the lungs aren't the cause of the problem. It's likely something else. So we're looking at metabolic acidosis or alkalosis, sorry. So we can cross out D because that's going to be respiratory acidosis. So it's not that, because that would mean that we have a high CO2. Oh wait, just kidding. Yeah, okay, yeah. No, we're looking at acidosis. So this is acidosis. This is also acidosis. So we can cross that one out. And then this is also acidosis. So the only one left is acute vomiting. And acute vomiting is going to cause metabolic alkalosis because of a loss in protons. Yeah, so that's how you would come across this answer. This is the only one that causes alkalosis, so it's really easy to like just rule everything out to answer that one. Okay, so the rule to differentiate between respiratory and metabolic would just to be look at both the bicarb concentration. So if bicarb is high, we know it's metabolic. If it's low, we know it's respiratory alkalosis. And then also compare that to your CO2 levels. So they will likely both be abnormal, but one of them is going to act as the compensatory mechanism, and the other one is going to be the cause of the problem. So it's really just differentiating between the two. That's like the trick in being able to understand what's going on and rule things out. Okay, so for question number 3, pH is low, so we know it's acidosis. Looking at our CO2 levels are high, which indicates it's likely respiratory acidosis. With respiratory acidosis, our CO2 levels are going to increase. Our bicarb levels are just slightly elevated, which tells me that this might be slightly elevated due to a compensatory response in the kidney from respiratory acidosis, but it's not like too high, so it's hard to call that the root of the cause. So that's another way to rule out metabolic acidosis. So it has to be respiratory. Looking at our oxygen levels are low. So this is another way to confirm the fact that we're looking at something likely due to hypoventilation. So oxygen is low, CO2 is high, that's going to occur in hypoventilation. So we know we're looking at respiratory acidosis. The only one here that leads to respiratory acidosis is D. So D is the correct answer. And lastly, another acidosis, because our pH is low. We look at our CO2. Our CO2 is also low, which would make you more alkalotic. So right away, we know that this must be the compensatory mechanism. Our O2 is within normal limits as well. So another way of determining that we're talking about metabolic acidosis. And bicarb is significantly lower. So it's supposed to be 24, but it's dropped down to 15. So that's another way of assuming that we are talking about metabolic acidosis. So now we have two causes of metabolic acidosis, either diarrhea or salicylate poisoning. So in order to differentiate between the two of them, once again, we have to look at the anion gap. Oops, that keeps happening. Our anion gap is high. Oh my God. Okay, our anion gap is high. It's 25. Our normal range is 8 to 16. So right away, that tells us that we have an increased anion gap. And one of the causes of those are salicylate poisoning, diabetes ketoacidosis, lactic acidosis. Other toxins can lead to that. So he's not going to give you like salicylate poisoning and diabetes ketoacidosis because that would be unfair. But he will either give you like something like salicylate poisoning or something like ketoacidosis or something. He'll use either or. Okay. So does anyone have any questions about those four or need me to like run through them again? If not, I mean, I can stick around for any additional questions or if you want me to go through any of the other kidney material. Again, I can do that. So he doesn't post the, I'm going to post my slides, I guess, right? Yeah, I'll post these slides later today, but he's not going to post his answers, I don't think, because they are very similar to the test questions. So I'll post these if you guys need them. And I like double checked with last year's data to make sure that my answers were correct. So I wasn't like totally getting it all wrong, but I'm pretty sure these are correct. You're welcome, guys. Oh, yeah. So the question about it was the anion gap question for a moment. Let me see if I can pull it up real quick. Okay, I think it's this one. So the question was, after the child was taken to the ER, blood work found decreased levels of bicarbonate in the patient's blood, which of the following was likely also discovered by his blood work. So ammonium chloride is actually going to have a normal anion gap, like HCl would or diarrhea. So that is the change to this. So the correct answer would have been normal anion gap instead of elevated concentrations of phosphate. Sorry about that, guys. You're welcome.
Hey, Phoebe.
Yeah.
I had a question, but it's like way back in lecture 3, where like you have it in your notes about the like thin descending and ascending, and so you have like increased osmolarity or osmolality, and I wasn't sure if like there was a difference when you're talking about this.
Oh, yeah, no, I think they're interchangeable because like it's just a different way of measuring the same thing. So one is like moles per kilogram and the other one is moles per liters. So it's the same thing when you're talking about molarity versus molality, at least for our purposes.
Okay, so then like when we make that distinction is going to be more so in like questions from the first lecture that like he explicitly talked about the difference between them.
Yeah, if he does ask you about that, I feel like it's too Gen Chem for him to like explicitly ask you about that. So if he does use it, he'll probably use molality, I think, is his preference. I think he just wanted to like, make sure you guys understood the difference from Gen Chem. So I wouldn't worry too much about it because it's gonna yield the same number, I believe.
All right, sounds good. Thank you.
You're welcome. I'll stick around for like, I don't know, 2 more minutes in case anyone thinks of any questions. And then after that, I'll end the meeting.
I also had another question about his questions. Do you think that he's going to give us the normal values, or should we probably know them?
Yeah, so I memorized the normal values. Like I remember memorizing them because I don't remember if he gave you the normal values, but I think just to be safe, you should, especially since his practice questions don't refer to the normal values. So I'm assuming I remember looking at a question on the exam and it was literally exactly like this table and like with the same kind of answer choices, but like obviously different numbers. And I can't remember if he gives you the normal values or not. I feel like he doesn't. So he kind of wants you to know, like, memorize just these normal values. Just know them like these 5 normal values and you'll be fine. It's not that many to remember, I don't think.
Okay, no problem. And do you have like your answers posted on Canvas?
I will post them later today. Yeah, sorry.
Okay, no, yeah, I couldn't find them, so I don't.
Yeah, I meant to post them, then I forgot, and I figured I'd just go over them and then post them in case like I got anything wrong, and then you guys call me out on it, and then I'd have to like remove them and then repost them. So I figured I'd just go over them first.
Okay, and then also, can you make one of these questions your practice exam question?
Yeah, I already made one like it. So yeah,
Okay,
one step
perfect.
ahead of you.
All right, cool. Thank you.
You're welcome. Alrighty, guys, if there's no more questions, I'm going to go ahead and end the meeting, and I'll talk to you guys on Friday. Feel free to send me emails if you have any questions about the material. Good luck on your histo exam. Bye, guys.